welcome to Basel and welcome to Sydney, which I think is fantastic because I'm from Australia and it's, Sydney's a great place to be. I was there in December. And what I'd like to say is that it's been exciting over the last day because we had an event yesterday and also today to see how exciting it is to meet people in real life. So given that, we'll be moving quickly through the pitches and I hope to give people a beer in their hand by a little after 7 o'clock. My name's Doug Hagstrom. I am part of the INCATE team and I am responsible for communication and the process for INCATE. And my partners in crime are Helmut Kessman, Silke Alt and Florian Kloss, who are respectively from the NCCR Anti-Resist and Uni Basel, DZIF and Infect Control. And they have a great amount of experience in the industry and a connection and network, which is what we try and use in INCATE to help. And the first thing about INCATE is we're about engagement and engagement with a community. So today is actually your chance to get involved and ask questions and also be doing the voting as well. So the audience here is our jury and you will be voting. So the way we're going to do this is with an app called Mentimeter. So if you have any questions for the companies as we go along, please enter them there. And it will also be where you will vote. So that's the logistics out of the way. I'll give you a very brief introduction into INCATE and the challenge we see. The challenge we see is that there's lots of great innovation. There could be more, but there's already quite a lot that's happening in universities and academic institutions where there are great innovators, and they're also happening in small companies as well. And they need to get across this gap to the industry. And I was excited to talk to someone today who said, this is so different to another conference because there's actually both sides here. The AMR conference has both academic people and it also has the industry here. And that's why as INCATE, we're excited to be here and part of this network and conference. Because what we at INCATE do is try and help innovators bridge that first gap amongst the many gaps there are on the way and points of failure. But we try and get them across that first gap to begin the journey. And we try and take that innovator across that gap with advice, community and funding. So advice is connecting to people we think who've had experience, have done that before, and it's connecting also to our partners and other people who can give that advice. That's part of that is a competitive selection process, and part of that is actually helping everyone we can. The second is a community, so we want to work together with people and bring innovators together to meet each other and bring people who want to help innovators to get a chance to actually give back and share the knowledge they have. And then we also provide some funding, a small amount of funding at first and then more over time. And we're also trying to be non-dilutive. Oh, we are non-dilutive. That's pretty simple. You just don't ask for anything. So what we actually do is, like I said, we provide advice and community to as many people as we can. And then as we work with groups, we then decide together with them to make a pitch for stage 1 funding, which is 10,000 euros, and then more detailed coaching and advice, and also work to develop a future funding and business plan. And then after a number of months of working together, we would invite them to pitch for a larger amount of funding, which will be a more detailed selection. We are not for people. We are a partnership of founding academic members, industry partners, so we're actually inherently bridging that divide between academia and industry. And then we have a number of supporting partners who are interested to support as we go along. And because we are a partnership, that's one of the reasons that inspired our jury choice today. So what's going to happen today is there'll be 10 pitches. There'll be a 4-minute pitch, about a minute for questions. If they're amazing questions, maybe we go a little bit longer. But otherwise, we move on and get closer to beer. And then right at the end, there'll be audience voting. So don't try and vote until then. But when the last pitch is done, we will then do the voting. I still see no questions from the audience. So we can get started with the pitches. The pitches will start with Invitrix. You see the order here, you see the order on your run sheet. And then, as I said, we can have questions directly in the app. So I'd like to invite Patrick on stage, who will give our first pitch, and we can swap over to the first pitch. Good luck, Patrick.
Thank you. Thank you so much for having me today. My name is Patrick Rossmann, and I'm one of the co-founders of Invitrix. Invitrix's goal is to remove all technological barriers that are currently limiting us to unlock the full potential of phage therapy. Phages are viruses of bacteria, and as a therapeutic, they will kill only those bacteria that actually make you sick. This is a clear therapeutic benefit, but generating broadly effective phage therapeutics is still a challenging topic. One of the problems is that we still need to rely on a method to produce phages that has not evolved for 100 years when phages were first discovered. We still need to rely on producing phages within cultures of pathogenic host bacteria. This in itself can be challenging, but a real bottleneck here is that because phages are so specific to bacteria, we will need separate production processes for every pathogen we'd like to target. This makes it extremely difficult to scale, and we solved this. We developed the worldwide first universal technology to synthesize arbitrary phages completely in vitro, completely in a single system, and completely independent of its original host. You can think of the system as an artificial generic bacteria that can take phage DNA encoding phage proteins and translate it into a functional phage. And we already demonstrated that this works for around a dozen of phage species across both gram-negative and gram-positive bacteria. This powerful approach can solve two key issues. Number one, scalability, because obviously our cost structure scales with the number of phages we'd like to produce. Number two, the emerging trend of phage engineering. Because we already work on the DNA level to produce phages, we can very easily modify its DNA and equip phages with new therapeutic mechanisms, such as CRISPR-Cas9 to specifically cut out antibiotic-resistant genes. Our goal is to develop this cure technology further into a generalized platform solution that will serve as a central phage factory of our company for phage identification, modification, and production. To achieve this, we already acquired more than 2 million of non-dilutive capital. We're able to file for 3 patents, and we're filing more. We verified freedom to operate with a patent firm, and we bounded the entire background and foreground IP exclusively to us. I think this puts us in a very strong position to create a business model in which we can fully capitalize on Factory to build a pipeline of our own drugs, but also to co-develop and license out additional assets with other biotech companies. In fact, we're already using Factory to develop a lead product against a chronic infection in a highly priced segment for which we are hoping to get our first in vivo proof of concept data by summer this year. And while there are around at least 10 other great phage companies around the world, we believe that our technology is so unique and complementary that most, if not all of those other companies could be extremely valuable partners for us rather than competitors, because we can help speed up drug discovery time by months, if not years, and benefit from their clinical experience in return. And I'm certainly not here to deliver the best phage pitch in history, but I'm here to compete for our team for the prize, because this could be the missing piece to help us file for a key patent that can enable an entirely new class of phage therapeutics using our competitive phage platform technology Factory. Thank you very much.
All right. I have one question.
Wait,
What is
well-
your current production limit in terms of scale and time to deliver?
That's very difficult to answer within 30 seconds because we still are not at a GMP scale yet. But we can produce titers of phages that are industry competitive. So we talked with a couple of contract manufacturers, and the titers that we can generate are the highest that we find in industry. We can produce titers of 10 to the power of 12, but at the same time, our endotoxin levels are so low that purifying this product, you lose really a little product of that original batch.
Okay. And one last question. How will you manage your own programs while you're providing services to competitors?
Okay, yeah. So our goal is we're currently focusing on expanding our platform and proving that we can generate specific mechanisms of action, different mechanisms of action with different phages. And at the same time, we're fully focused and committed to one asset that we're developing. All the co-developments we will do once we hit the preclinical stage that is so late that we can safely shift focus.
Okay. Thank you, Patrick.
Thank you.
All right. Our next presenter is from the UK, and it's Amprologics.
Hello, everyone. I'm Gordon Barker. I'm co-founder of Amprologics. We're an antimicrobial peptide company. So here's the AMR crisis in just a couple of numbers. Depending on whose numbers you look at, you believe costs between $1 and $3 billion to bring a new therapeutic drug to market. And yet, about $280 million is the average sales cumulatively for a new antibiotic in its first 8 years. And it's really hard to make those numbers work. So at Amprologics, what we're trying to do is bring that number down and change the calculus. We're confident we can bring new epidermicin antibiotics to market for about $30 million, just under. How do we do that? Well, for a start off, we're playing within the rule set of peptide chemistry. It's much cheaper and faster to systematically change peptide sequences than it is to dabble in the dark arts of small molecule medicinal chemistry. And we're also leveraging clever technology and new business models to further reduce the cost of development. At the heart of it is this. We take all our SAR data, everything we know about the peptides, and we put them into a machine learning algorithm. It's the same AI that sits behind the deep fake technology, the videos and photos you might have seen. And we then rapidly synthesize and test those peptides that the AI suggests. And the fittest peptides from our bioassays get fed back in an accelerated evolutionary process. So we're spinning round and round this as quickly as we can. Currently, this is aspirational. The robots we're using are those really programmable but slow ones called postdocs. But we've established a sort of proof of principle. And there's other people playing with these tools. And certainly, as was mentioned just after lunch, antimicrobial peptides come slightly loaded with negative expectations. But what's different about us? Well, we had a really good start point. The peptide space we're exploring is nearly infinite, and yet we got lucky. And our start point is this molecule. It's isolated from a human skin condensal, so it's co-evolved with humans over millennia, and it's really safe around human cells. Not only that, but it's a novel structure that's really tolerant of modifications. You can play with this sequence, and it doesn't affect how it kills the cells. You can even tag things to it and retain that activity. There's no known resistance mechanisms, and we've tried to make them and failed. So it's a pretty good platform. Does it work in the real world? Well, this data is from a CRO, probably the leading one in the infectious space. And basically, it shows that our lead compound, the core of our program, is about as efficacious after a single dose as 5 days of twice-daily dosing of the current gold standard. So it does work in the real world for all that it's a peptide and is susceptible to peptidases and all those other things. In terms of the development cycles, this is just a quick snapshot. The upper table shows how we're using the AI to dial down the MRSA activity that we just showed you previously and dial up activity for Acinetobacter instead. And then on this separate table here, we are using the AI to optimize some fusion derivatives that are going to be more active in community-acquired pneumonia, which is where we want to go, having demonstrated their nasal activity. So that's where I wanted to stop. We've got an exciting platform. We just need seed financing to realize its potential. I'll answer any questions if there are any. Thank you for your time.
Thank you, Gordon. If people are old school, you can also put up your hand. Or we do have a question here. I think this is a traditional one. You should be able to answer. Immuno-relevant side effects? Question mark.
Really good question. So we've not done all the in vivo tox testing that we'd like yet, just for lack of funding, but we've got some pseudo-allergy data with human skin models, and there's a surprising lack of activity there. It doesn't seem to interact with any of the known cascade targets for that. So we're investigating why it's so good, but it's probably this co-evolution that it's had with the human species for millennia. So it's good there. We've got some decent in vivo tox data from the nasal model where we've looked to see if we see any negative side effects in the animals that we've dosed, and we don't. So again, we've drawn a blank there. More work's needed to be done, but we've got good proof of concept.
Okay. One last one. Can machine learning predict chemical modifications of amino acids?
We're not doing non-natural amino acid modifications yet. We don't feel the need to go there, frankly. But yes, the pattern recognition kind of algorithms that we're employing they find connections that the human brains don't, and take in a whole raft of data, spot patterns and structural features that we don't see, and then spit them back out again into novel arrangements. We're surprised with what this algorithm spits out. I'm absolutely confident that it could handle anything that looks like a peptide.
Okay. Thank you very much, Gordon.
Thank you.
And just so for information, some of you had a few more questions that came in a little bit late for the first two pitches. That's why we have networking afterwards. So please make contact and have a good chat and see how you can help these young innovators. I'd like to ask our next innovator, who I'd also describe as young at heart, Frank, welcome on stage.
Thanks a lot. INCATE, Doug, and also the conference organizer and Fossios for this pitching event. I would like to present the anti-infective company Salmo to you. So we are fully dedicated to AMR. We are focusing only on targets or new chemical modalities which have been proven in vivo already. As mentioned, new targets or also established target, but then a new chemical modality and a new mode of action. And we also have a focus on this in the resistance. I'm very happy to present to you really a highly motivated team of founders and experienced managers and experts in the microbiology business. Adam Bailey, located in Montreal in North America, and also the founder of the company, Philipp Knechtle. And also in the team we have Professor Süßmuth, chemistry expert, and life science entrepreneur, multiple startups, and now working for a headhunting company, Markus Hofer. And also with our scientific advisory board, I'm also very happy to have the key opinion leader, Mahmoud Ghanum from the Case Western University in Cleveland on board, who helps us with the antifungal and the antibacterial assets and the microbiome. And then we have other support functions. Let's go into our pipeline. So we have 3 assets currently running. The most advanced is the SLM500 against antibacterial infections. We have acquired an exclusive worldwide license from the Technical University Berlin and partnered this project with the incubator from iCuris, Anti-Infective Cures, where we have really good support. Then our second antibacterial target is very early. It's against gram-negative bacteria, very early in the lead finding status, and we would like to perform the lead optimization. The next milestone, and then also we are one of the only rare companies which have also an antifungal asset against systemic infections, where we filed last year a patent application, and we would like to perform the lead optimization and the in vivo pharmacology. So the investment opportunity is over $4 million or euro or Swiss francs over all 3 projects and different stages, and you see the from the timeline, we would like to reach with the most advanced, finish the lead optimization stage, and provide 3 analogs for further IND profiling. The antifungal project is then the next more advanced project where we need additional lead optimization and in vitro and in vivo characterization, and as mentioned, the second bacterial target is very early. Let's go into the science of the SLM500. So we are talking about lead structure called albicidin, a natural product, and it is active against the well-established target of gyrase, where a lot of fluoroquinolone type antibiotics are already on the market, but facing high resistance. It is a new chemical modality, complex structures, amenable by synthetic chemistry in a modular way, and also scalable. The resistance rate is quite low, 10 to the minus 11, and we have also performed in the next slide in vivo proof of concept, and a very excellent patent position with 2 granted patent applications and 2 pending applications. I missed the minute alert.
All right. Yeah. We do have one quick question. Host toxic effects?
The cytotoxicity on the cytotoxicity safety margin is 1 to 1,000 on the lead structure, and on the antifungal structure it's also in the range of over 1 to 100. But there, if we clearly need more pharmacologically improvement.
All right.
Yeah. So
thank you, Frank. Yeah. And if any of you want to follow up with Frank Science, then catch up with him at the networking. The next group on stage has come from Norway, which is very close to my second home, Sweden.
So thank you for inviting me to Basel first time. I actually almost went into the wrong country coming into this country. We are working we're working at Biotech, and we are looking at beta-lactamase inhibitors. And there we go. This area of AMR was in the interest of Professor Rongve at the University of Oslo. And we set up a team last year of these people here. We attracted €5 million and got started with our non-clinical program with our compounds. As you all know, or most of you at least, beta-lactamase inhibitors are combined with antibiotics to protect them from resistant enzymes, making them more sensitive in the patients. This is exactly what our 2 compounds are doing. So APC-148 is a selective MBL inhibitor in the non-clinical stage. The APC-247 is a combined MBL and SBL inhibitor. We have earlier proven that APC-148 is quite effective in increasing the sensitivity of meropenem, and this has been published a couple of years ago. So what we do today is we are validating these results in a much broader number of isolates, and of course also using more resistant isolates to prove the efficacy of this compound. The next generation beta-lactamase inhibitor is APC-247. We just received Eurostars funding for initiating this non-clinical program, and we have high hopes for this compound that is more broad spectrum in the sense of enzymes. The company also has some activities, some academic activities in the area of resistant fungi and tuberculosis. The company has just recently been in discussions with the agencies, FDA, EMA, and the national authorities, and we are quite satisfied with those discussions and feel that we have a very good validated plan for our preclinical program, taking us into an IND or CTA with our lead compound, APC 148, by next year. We know that there are competitors out there. We feel still we are in a very attractive space when it comes to the product. And we also know that half of these competitors will be non-successful. We hope to be on the other half. And still we see that in the future there will be a need for many of these products, and we don't feel that's uncomfortable to be in a competitive space like this. We all know that this is a big problem. This is a recent outbreak of MBL producing pathogens in Portugal. Both our products are addressing this issue. And with these words, I will just say thank you so much for your attention, and please, any questions if you have.
Right. Thank you, Bjorn. Sure. So we're now 4 pitches in, and the 5th pitch, I'm looking around, is Thomas Hesterkamp from Hhv Biotech.
Good afternoon. It was Rolf Müller, Jenny Herrmann, and co-workers from the Helmholtz Center for Infection Research who first isolated and characterized the anti-infective drug class of the cystobactamids from extracts of the Myxobacterium Cystobacter velatus. And since we have optimized this drug class by chemical means, deeply profiled biologically and pharmacologically, and extensively patented this drug class. The cystobactamids hold a potential as broad-spectrum antibacterials. So we have mapped here in green those bacteria identified as global killers and who fall into the activity spectrum of the cystobactamids. However, we chose to initially focus our optimization efforts on carbapenem-resistant Acinetobacter baumannii. CRAB, shorthand, is a global health threat, an acknowledged health threat. It's a gram-negative bacteria causing nosocomial pneumonia and wound infections. It has a high disease burden in Southeast Asia and other parts of the globe. There is an excess attributable healthcare cost associated with the treatment of the drug-resistant infection. In terms of background, standard of care poorly defined. According to Infectious Disease Society of America, the standard of care might be a high-dose Amsul. Unmet medical need patients would get polymyxin or colistin, which is toxic, or cefiderocol, which suffers high resistance rates. Guidance is for combination therapy, and there was one late-stage pipeline product. So the cystobactamids act as dual topoisomerase 2 inhibitors in this bacteria. So phenotypic studies indicate DNA damage, frequency of resistance formation is rather low, and the mechanism of resistance is on target, but outside the quinolone resistance determining region of the fluoroquinolones. In terms of cidality, the mechanism is rapidly cidal, as you can see here, and we can effectively suppress any regrowth within 24 hours at multiples of the MIC concentration. In terms of MIC distribution, the cloud of the cystobactamids here show a unimodal MIC distribution curve with MIC 90 values in clinical isolates of regularly below microgram per milliliter. In terms of in vivo efficacy, we can suppress bacterial burden in blood in a mouse septicemia model down to substatic levels, likewise in kidney, while in spleen at fairly high inoculum, we drive down bacterial burden to static levels. In terms of lung distribution, we can show that the drug passively permeates to the epithelial lining fluid of the mouse lung, and RTI models are in progress. In terms of mid-stage planning, we're planning to have our candidate selection event by the end of next year. To that end, we're seeking seed finance in the order of 3.6 million euros. In terms of team, it is Jenny Herrmann and myself who will take operational roles in the company. We're currently seeking to add to our team, so we're recruiting, and it is Rolf Müller, Mark Brönstrup, who are our founders and advisors to the company. In terms of investment opportunity, we license intellectual property from the Helmholtz Center for Infection Research. We currently seek to raise seed finance. We see a great opportunity to follow up projects from the Hhv pipeline, and we have been selected as one of 4 in the first round of the INCATE selection process. Thank you.
We do have a couple of questions. Any profiling done on the human microbiome strains? Exclamation mark question. Or just the IV administrative route? Also, exclamation mark question. Sadly, we can't have emojis, I don't think, but you can try that in the next one.
Not specifically, no.
No, not specifically. An easy question. Manufacturing of the product, what does that look like?
We have done that straightforward, so we regularly procure building blocks from CROs, and this is straightforward.
Great. Thank you, Thomas.
Thank you.
As you heard, we're halfway through, and to break it up a little bit, the 6th group will be coming forward with 2 presenters. So, SmartBax team, welcome on stage.
Hello everyone, we are Magdalena and Robert from SmartBax. And we at SmartBax develop a new generation of antibiotics against multi-drug resistant bacterial infections. And our compounds are characterized by a novel multi-targeted mode of action, which has already shown to be successful in vivo in proof of concept study with our hit compound. Our current focus is on MRSA and vancomycin-resistant enterococci, and a particular feature of our compound class is that we can show strong activity against persisters and biofilms. Furthermore, we're also setting up a unique high-throughput screening program against our targets to come up with new and independent lead structures. Biofilms and persisters are a really challenging subpopulation of bacteria that can cause severe infections such as endocarditis, bone and joint infections, or periprosthetic and catheter-related infection. And it's really our goal in SmartBax to develop novel small molecule antibiotics against these particularly difficult to treat bacteria. We're doing this with a unique dual-targeted approach that acts via 2 independent enzymes, MENG and SBSB. MENG is essential for the biosynthesis of the electron carrier minakinone, and when we block this pathway, the bacteria are under respiratory stress. At the same time, we are able to not inhibit but actually activate the enzyme SBSB, which is a signal peptidase in the secretory pathway of the bacteria. And we believe that this secretion enhancement causes an increased secretion of autolysins, and these will then disrupt the cell homeostasis and slowly degrade the cell wall. Overall, this mechanism of action leads to some quite favorable properties because we do not see development of resistance with our compounds within 30 days of serial passaging. And when we look at their MIC90 distribution, over 100 different staphylococci and enterococci of various clinical resistance patterns, we see a very sharp distribution of MIC. And this is indicative that this is really a mode of action that is different from common resistance mechanisms and really a new mode of action. We can also see, and we see this throughout our whole compound library, that we are able to kill persister cells and also eradicate formed biofilms.
Our goal is to build a sustainable antibiotics development company. So we have a 2-sided strategy. First, we will further develop our front-runner programs, and also we will set up a unique high-throughput screening, a targeted high-throughput screening, to also develop new ideas for our pipeline. We are currently privately seed funded and are also very lucky to be selected by INCATE Stage 1. And will raise further funding throughout this year. Our team combines expertise in chemistry, microbiology, business, and also industry. So we have with Stefan, Robert, and Franziska, we have chemists on board who have a track record in discovery of antibiotic drugs. With Elizabeth, our microbiologist, we gather a deeper understanding of our targets, and we are also very proud and happy to work very closely with Peter Hammann and Rolf Müller as scientific advisors and also industry advisors who support us with their knowledge. So we all gathered here today, and we are here today because we want to fight AMR. At SmartBex, we have a novel multi-targeted mode of action with strong activity against persister cells, and we have a strategy also to build a new backbone pipeline of new ideas. So, and furthermore, very important is we as a team are ready to fight this challenge. We are at the very beginning of our journey, so support us on our way to novel antibiotics. Thank you.
We've got a couple of questions. Toxicity. What about toxicity, especially the menaquinone targeting part? Scientists, not the CFO.
Well, the advantage about these targets is that they do not exist in humans. Menaquinone humans need, but they take it up as a vitamin, so we don't have like an analogous enzyme that could be targeted. We're screening constantly, of course, the toxic effects of our current compound pipeline. We see fairly little off-target profile in the seroprofiling, but definitely this is something that we have a look on.
Okay, great. A last one, science one. Have you targeted various persistence mechanisms?
Well, I guess the question is also like how the persistence mechanism exactly is targeted. Our hypothesis is that it's really also the overactivation of one of the enzymes is part of this activation mechanism that then is also able to address bacteria that are actually metabolically dormant. So that is our theory, how we basically achieve this mechanism of persistence that we address.
All right. Thank you. Thank you very much. And on to our... Thank you. Now on to our seventh presentation. It's taken a while, but we've finally got to some diagnostics. Welcome on stage, Melanie.
Thank you for the invitation. My name is Melanie Schepers. I'm with Sefunda. We are a diagnostics company based in Muttens. It's like 3 kilometers down, I think, that way, right in Basel. Yes, what are we doing? So that's Basel in summertime. COVID is over, or at least declared over. People are enjoying themselves, and we are asking ourselves, what could possibly go wrong? Well, we know that infectious diseases are on the rise. So I think we have like the numbers triplicated since the 80s. And we have, especially for the sexually transmitted diseases in the UA, we have a rise of 56% between 2015 and 2019. With gonorrhea on the verge of being untreatable. So when we started our product, the CDC still recommended a certain antibiotic treatment, and they changed that in January 2021, so that we are now on the last resort antibiotic and working in the direction of being untreatable. So again, from an industry perspective, this came up, I think, on March 24th at the World TB Day from GARD-P, that actually the bad news is there are no antibiotics on the horizon anytime soon, since what we all know and what's discussed all around on this conference, this is not a business case for the pharmaceutical industry. But there's also good news. We have still 50%, for example, for all gonorrhea infections, are still susceptible, that's my beloved word, susceptible to efficient and safe alternatives like older antibiotics. But rapid diagnostic that include these kind of markers are rare. The industry usually focus on detecting CT/NG, but no resistance markers. These are key to get this information, to get access to these older alternatives as a way to preserve antibiotics. So the challenge is that the common testing strategy is usually too slow. Since everything is usually sent to a central lab, you wait for the results, and then they are not looking for antimicrobial resistance markers. And also what we need is a low barrier solution that's more decentralized, that's fast and easy to use. So that we have the additional possibility to get diagnostics everywhere. So our solution is that the Funda is developing a rapid diagnostics platform where we get information for the causative agent and the AMR in a single patient visit, built-in data collection, cloud capability as we call it, and what we are very much focusing on is that we have that on an industrial scale. Because what we learned during the pandemic is that a diagnostic solution is only then a solution if it's available at scale. Okay, so that's our platform. We are doing end-to-end run right now. So that's our prototype and our cartridge. This is a team, and we are dedicated to get this thing to the market. We are looking for some financing, which we can discuss later by a beer, obviously, because putting such a device on the market is actually very expensive. So thank you for your attention. Any questions?
An easy one, I think. Which different diseases have you tried and tested with your platform so far?
We are developing a CT-NG plus R plus LGV. I didn't mention that we also detect the LGV serotype for chlamydia. The next assay is going to be a very obvious one, a COVID flu RSV assay. That's in development right now.
All right. I think we have a few more questions. Your TRL number, do you think in TRLs?
Sarah, do you help me? You are the scientist on this one. I have no idea.
The technology readiness level, but...
Technology. Yeah. Somebody has to explain me what they mean. So what we are very much focusing on is really that we can produce at a mass scale. So we design for manufacturing is our focus, so that we can actually are able to put the design and the development on the market at a low price at mass scale. So that's the focus.
Okay, it seems like you've been following the questions directly. Cost per test, what's your target test price?
Well, if my wishes all come true, we are below $10, but right now we are higher.
Great.
But it's a secret. No, we are not that far away. But the thing is, production is really difficult, and you have to get down the cost. And we are really focusing on getting the plastic parts as simple and as easily to produce as possible. And somebody find me a chemistry providing company that doesn't cost so much, please.
Okay. It sounds like we've got some work in the networking.
Yep.
And thanks to COVID, maybe some of these costs will come down.
Hopefully. We will see.
All right.
Thank you.
Thanks a lot. Now, from Muttens to Holland, welcome Hhv Biotech.
Good afternoon or good evening, everyone. My name is Kim, and I am CEO of Hhv Biotech. As you probably noticed, I'm not standing here alone, but with this little one inside me, who is expected to come out in June. So thanks to the organization of the perfect timing of this pitch. Thank you for the opportunity. Now let's talk about Hhv Biotech. Our company's mission is to tackle biofilm-related infectious diseases. And thanks to my colleagues who already explained biofilm to some extent, biofilm plays a key role in AMR. It's basically a community of bacteria that are enclosed in a self-produced extracellular matrix that consists out of different components. The bacteria inside this biofilm can actually escape the immune system as well as traditional antibiotic treatment. If you look at these numbers, out of all microbial infections, 65% is biofilm related. And as much as 80% of all chronic infections is biofilm related. And yet, until today, no dedicated anti-biofilm treatment is available. Now let's zoom into a primary indication and how does this affect our patients. in the US alone, 10.5 million patients suffer from UTI each year. 1 in 4 will end up with CUTI, and of them, 20% will end up in the hospital. Now, what's very interesting, but also concerning, out of these hospitalized patients, over 80% received only 1 oral treatment, which shows the desperate need for new therapies in this space. Now, biofilm is one of the key reasons for CUTI. As you can see in this picture, the biofilm attached to the bladder wall is basically unreachable for common antibiotics, so they're ineffective. Also, the biofilm contributes to rapid spread of resistant genes and persister formation. And this all results in long-lasting infections that put a lot of burden on the patient and healthcare cost. Our lead drug candidate, Hhv shows promising efficacy against the 3 most prominent CUTI-causing bacteria: E. coli, Klebsiella, Pseudomonas. They all account for over 65% of the pathogens that cause this infection. Now, what's even more interesting is that our compound showed to not induce resistance, in contrast to existing therapies, ciprofloxacin, for this indication. We were founded early last year in 2021, and since then we already reached a couple of important milestones, including GMP manufacturing of our oral small molecule. We have enough material to finish the preclinical stage as well as the phase 1 stage. Also, we demonstrated no adverse effects, up to 100 milligram per kilogram in rats, also in mice, which we heard this morning, in our UTI efficacy models, where we expect data in the coming weeks. If it all goes well, we aim to be first in man, first quarter 2023. Our team consists of a very interesting mixture of biotech and pharma experts, microbiology scientists, infectiologists, and serial entrepreneurs with significant deal-making experience within this industry. Together, we're on a mission to heal biofilm-related infectious diseases, and personally, I'm committed to contribute to a pandemic-free world, especially for this little one inside me. Thank you.
First question, mode of action. What do you know?
Yeah, that was one of my questions this morning. We see in clinical relevant models very nice efficacy. We are currently unraveling the mode of action. What we do see is that compared to antibiotics in dose response curves in biofilm models, yeah, we have an effect when the bacteria are embedded in the biofilm, whereas the antibiotics do not. And we don't know exactly how it works.
Okay. Sometimes it's better not to know things. One question. Is the drug cleared significantly over the kidney?
Yes.
That was quick.
That's
good.
what we see in the urine, because we can find it back.
And then there's an unfair question, which I'll ask anyway, which is, how can we vote against a mother? Thank you.
You can't.
And thanks very much. Yeah. That quick reminder for the voting, you'll be able to choose your top 3, and that will be opened at the end of the final presentation. And we have 2 presentations to go, and the next presentation has come also from the Scandinavian direction, and Finland. Welcome on stage.
Right, so we are Hhv Therapeutics, and let's see. Oh, right, great. Yeah, so we see antibiotic resistance as the main problem of what we're facing. And quite soon after we developed the first antibiotics, resistance was already becoming a problem. And if you fast forward to today, we've exhausted most of the pipeline that we have. And it's even put a chokehold on the development of novel treatments. I guess you hear a lot about that here at the conference. And basically, what we were thinking and what we really wanted to know is how can we get around this? And that's the main focus of our company, is really to focus on how can we develop treatments that do not face this resistance, but are still effective in treating these pathogens. And we're going to show how this works in the case of cystic fibrosis, where we're focusing on the virulence inhibition. So very briefly, cystic fibrosis is probably quite well known. It's a genetic disease that causes the excessive production of slime inside the lungs. And this makes these patients very susceptible to bacterial infection. And they basically have continuing pulmonary exacerbations, and eventually they die from this. Very briefly, the reason why we decided to focus on this is there were very few treatments available for these patients. It's a very severe disease. And Pseudomonas aeruginosa, which is a key pathogen we're targeting, has a very high prevalence in this disease. So how it works, we basically target the virulence. And the molecules we work with have a dual functionality. And it targets both offensive virulence through binding of bacterial signaling molecules in its inner cavity, and defensive virulence capabilities through disruption of lipopolysaccharides in the bacterial outer membrane. Now, a very brief summary of some offensive virulence traits, which are biofilm and toxins, and we see a nice dose-dependent reduction of that. And similar to what some other projects have also shown here is that we see that it also eradicates already formed biofilms. So biofilms are really a major problem, particularly also in cystic fibrosis that makes them much more difficult to treat and clear with antibiotics. So basically we see that the treatment can effectively penetrate and clear these formed biofilms. And combined, why we think it's so interesting to look at virulence and virulence inhibition altogether is because it is really very effective against multi-drug resistant clinical isolates. So where antibiotics don't work, virulence inhibition can still be a very effective strategy. And overall, we see that it, as a standalone treatment, although it has no direct MIC, it is still a very effective way to treat bacterial infections in, for instance, lung environments. But what we see as the key problem, and the key problem we need to solve, is resistance. And resistance to antibiotics can develop extremely fast. So in a 14-day experiment, for instance, you can have resistant levels already in as soon as 4 days to standard antibiotics. And we think virulence inhibition could be extremely effective because we don't see the same resistance development. And basically, the treatments stay effective throughout this period. At the same time, you can also use it together with antibiotics, so it synergizes a variety of antibiotics. Here we show an example for cefepime, and in a combined administration, basically what it does, it reduces the resistance buildup also to the antibiotic. So there is a strong synergy there. In terms of what are we developing and how are we developing it, we're developing it as an adjunctive therapy, an inhaled adjunctive therapy to antibiotics. And some of the primary endpoints we're aiming for is improved lung function, FEV1, as well as a reduction in the severity of pulmonary exacerbations. So really, it's very different from standard antibiotics that all have a MIC value, so either bactericidal or bacteriostatic. So it's really, we don't have that, so it's a very big shift in terms of also thinking about infections and think about treating of infections, but we see it as absolutely necessary. We have identified basically a strong team of 3 founders, and we're planning to spin out from the university this summer. And we're planning to do that with an IP portfolio of 3 patent applications and 1 single granted patent. And basically, in the beginning of 2023, we're hoping to raise a 4 million seed round to basically take this forward, both in the primary indication into IND-enabling studies, as well as 2 different programs on sepsis and wound care. And that is us, and we're trying to tackle resistant infection through virulence inhibition. Thank you.
Thank you, Christoph. Do you know whether the virulence factors you are targeting are relevant for pathogenesis in CF?
Yeah, right. So it doesn't, it targets, that's a bit of a complicated question because it has direct targets that are virulence factors. So lipopolysaccharides directly in the signaling molecules that we target, yes. Pyocyanin is in the question. That's an indirect mechanism in how it works. So it doesn't directly target and sequester the pyocyanin, but it regulates the production of pyocyanin and other toxins.
All right. Thank you. That sounds like a beer conversation. Thanks a lot. We're now on to our last presenter, Laura Yates from Zurich, welcome, and a new modality, vaccines.
So, good evening, everyone. Thank you for still being here for the last talk of the last session of the day. I'm Laura Yates from Hhv Biotech, and I'd like to present to you our solution for beating antimicrobial-resistant gonorrhea with a novel vaccine. So, the concurrent rise of antimicrobial resistance and incidence of sexually transmitted infections is a dangerous combination. Over the past decade, cases of gonorrhea have more than doubled in many countries around the world. In the US, for example, this means an estimated 1.6 million new infections last year, of which half were antimicrobial resistant. And over the past 80 years, gonorrhea has developed resistance to all antimicrobial treatments. Already today, there's only one recommended treatment remaining, and resistance has been observed against this in multiple countries. Neisseria gonorrhea will continue to develop resistance against new antimicrobials, and we believe that in just a few years, untreatable disease and all of its consequences will be widespread. A better solution is urgently needed, a vaccine. Vaccines do not contribute to AMR, and they're entirely pathogen-specific, so they're harmless to the natural microbiome. Taking Streptococcus pneumoniae as an example, itself a serious AMR threat. Since the introduction of the pneumococcal conjugate vaccine, not only have millions of antibiotic prescriptions been avoided, but the levels of circulating resistance have actually significantly fallen. Vaccines even protect our existing antimicrobials. Our gonococcal vaccine is unique as it employs novel antigens that overcome the natural immune evasion mechanisms of Neisseria gonorrhea. Our highly skilled team combines global experts in gonococcal disease and significant expertise in vaccine development from the bench to the clinic. In fact, my colleague, Christina Alaimo, will be talking about our Klebsiella vaccine trial tomorrow. So for the gonococcal development program, high incidence populations mean that clinical efficacy may be demonstrated as early as phase 1. And our cost-effective production platform will also allow us to address global health markets. We have 3 established preclinical assays, including a serum bactericidal assay, which is an established correlative protection for Neisseria meningitidis vaccines. These 3 complementary assays have been used to evaluate each of our antigens individually, and we're now at the lead optimization phase. Strong performance in these assays clearly demonstrates a functional mechanism by which the vaccine can protect against gonococcal disease. The threat of untreatable disease will drive demand, and the success of the human papillomavirus vaccine paves the way for a new introduction alongside this now established vaccine. Sales of the leading HPV vaccine last year totaled $5.7 billion, so comparable revenue is anticipated. We are Hhv Biotech, a team of 35 highly skilled individuals with significant expertise in vaccine development. We have an experienced management team, and our board includes many entrepreneurs who've been involved in previous successful startups, including the serial entrepreneur Philippe Drault. We also have a large number of international partners and collaborators who strengthen our team. We're seeking funding to support the development of our proprietary pipeline, which includes this, along with other products at different stages of development. So if you're interested, please get in touch. Thank you to the organizers for the opportunity to speak here today, and to you for your attention.
And 3 questions for you. How do you select promising antigens?
So these antigens were developed by a collaborator. We've in-licensed them, and these preclinical assays really give us a strong feel for the potential for them to truly be efficacious in protecting against gonococcal disease.
Yeah. A bigger question, maybe, to describe, acceptance issues for vaccination. What do you think about that?
That's a good question. The human papillomavirus vaccine, the success of that, I think, has really been beneficial in changing attitudes towards vaccination, specifically in this space, the use of vaccines against sexually transmitted diseases. And I think it's also important to bear in mind that as the threat of untreatable disease gets ever closer, this is not just a vaccine against gonorrhea, but it's a vaccine against infertility, against chronic pain, all of the other consequences that come from untreated gonorrhea.
All right. What about price per dose? Do you think something like the HPV?
No, I think we can afford to produce at a significantly lower price than the HPV vaccine. As I mentioned, we have a very cost-effective production platform, and while we're not at the stage yet of putting a price per dose, we really believe that we can make this substantially more affordable than the HPV vaccine, which is a very expensive vaccine.
All right. Thank you, Laura. Thanks very much. Okay, we're now over to the audience, and it's your job to do the next stage, which is actually to vote. You should either scan the QR code or go to menti.com and 7248451 and choose your top 3 companies. So this is your chance to play investor and funder. You'll have 2 minutes of thinking time, so there'll be some thinking music, and then we will have a prize ceremony. Could Mr. Werner from Fossius come forward as well in the break? All right, that is exciting. Even more exciting to see the results come in live. I would like to invite on stage Joshua Werner from Fossius, keeping the check the other way around, hiding the check, keeping the suspense going. So while we've got everyone's attention, Joshua, Fossius has chosen to help out today and support INCATE. Why does Fossius support things like INCATE and the innovators themselves?
Fossius and Partner has a strong focus on AMR and especially on IP, and we are happy to support startups in this field because we think there is a strong need for a clinical need, and that we think that is currently unmet, and we would like to support in this area, and we are happy to sponsor the prize today.
All right. Thank you very much. We now have the technique ready, and the winner is Limatech and laureates. So welcome on stage again. All right, plenty of flashes. And Laura, would you like to come over here briefly?
I wasn't ready with an acceptance speech. So yeah, thank you very much, everyone. I'm really delighted to see that vaccines are being recognised as an important tool in the fight against AMR here, because I've heard so much during the day about antimicrobials, and I really think it's important that we look broader. There are lots of different approaches that we can take, and yeah, pleased to see vaccines getting some recognition.
Congratulations. Maybe next year we'll have a bit of vaccine competition. All right. Thanks, everyone. Thank you for coming to the the pitching competition today. Thank you for staying engaged. Thank you to everyone who voted, got engaged. The work starts now, actually. It's about helping this group of 10 people to keep going and keep working. So please engage with them during the networking session, and we look forward to seeing you again next year. Okay, thanks a lot.